I had the privilege of presenting data from the first-in-human phase 1 study of PLN-101095, which is a first-in-class dual AV-beta-8 and AV-beta-1 integrin inhibitor, given both as monotherapy and also in combination with pembrolizumab in patients with advanced solid tumors refractory to immune checkpoint inhibitors. The whole scientific reason for this particular trial is that in response to sustained immune activity, solid tumors utilize integrin AV-beta-8 and AV-beta-1 activation of TGF-beta to suppress and escape immune control. And therefore, PLN-101095 was designed to potently block integrin AV-beta-8 and AV-beta-1 driven activation of TGF-beta locally in the tumor microenvironment. And this is different from past strategies of systemically targeting the active TGF-beta cytokine, the TGF-beta receptor kinase, or specific isoforms of TGF-beta. And PLN-101095 selectively enhances T-cell interferon gamma effector function. It reduces fibroblast activation and fibrotic tumor stroma, and therefore inhibition of integrin AV-beta-8 and AV-beta-1 blocks the activation of TGF-beta to really reduce immunosuppression, leading to a new or reinvigorated cancer immune response. And overall, by inhibiting TGF-beta, PLN-101095 shifts solid tumors to a high interferon gamma signature and an immune checkpoint inhibitor responsive state. And that provided the rationale to proceed to this phase 1 open label dose escalation trial in patients with solid tumors with prior immune checkpoint inhibitor resistance. All patients were mandated to have prior exposures to PD-1 therapies with documented disease progression and were required to have primary or secondary resistance by SITC definitions. The primary and secondary endpoints included the assessment of safety and tolerability and also pharmacokinetics, whilst the exploratory endpoints included antitumor activity and changes in blood-based biomarkers. We explored different dose levels, ranging from 250 milligrams BID up to a top dose of 2,000 milligrams BID of PLN-101095, given as monotherapy and in combination with pembrolizumab. Overall, we treated a total of 16 patients during dose escalation. It was well tolerated, and the most common treatment-related adverse event was grade 1 to grade 2 rash. There was one treatment-related rash that was reported during the monotherapy period, but otherwise the rash was primarily observed within 2 days of starting combination therapy. In terms of pharmacokinetics, the PK profile supports continuous pharmacologic inhibition with BID dosing at steady state. And importantly, anti-tumor responses were observed in patients with secondary immune checkpoint inhibitor resistance, including 4 patients with iRECIST responses, including 1 complete response, 2 partial responses, and 1 unconfirmed partial response. In immune checkpoint inhibitor secondary resistance patients, the overall response rate was therefore 30%, with a disease control rate of 60%. Overall, we observed clinically significant and durable responses in 3 out of the 4 iRECIST responders at doses of 1,000 milligrams BID of drug or greater. And the median time on treatment was 19 months for the 3 confirmed objective responders who had an average maximum target reduction of minus 89%. Interestingly, we also observed that increases in interferon gamma during monotherapy may actually act as a potential biomarker of TGF-beta inhibition. We saw that clinical responses to PLN-101095 was associated with elevated plasma interferon gamma and PD-L1 levels after just 14 days of PLN monotherapy. And this will be studied further in our dose expansion cohorts. So to conclude, PLN-101095 was generally well tolerated in this dose escalation part of this phase 1 trial, with no new safety concerns emerging when this integrin inhibitor was combined with pembrolizumab. Importantly, early signals of antitumor activity were observed in patients with immune checkpoint inhibitor secondary resistance, with an overall response rate of 30% and a disease control rate of 60% when treated with PLN-101095 plus pembrolizumab. Circulating interferon gamma may be a potential biomarker for early prediction of treatment response, and dose expansion cohorts are planned for initiation in the second half of 2026. These data for PLN-101095 plus pembrolizumab suggest the potential to meet a high unmet treatment need amongst these patients with secondary immune checkpoint resistance with no new safety concerns. Thank you very much.